Welcome to my YouTube channel. In this video, we will learn about how we can perform RNA-seq analysis in Google Colab by using machine learning. First of all, we will discuss about what is transcriptomic analysis, and then we will talk about RNA-seq. Transcriptomic analysis is basically the study of RNA transcript, and this analysis helps researcher to understand the gene expression and different regulatory mechanisms. Now, what is RNA-seq? RNA-seq is used in transcriptomic analysis to capture and quantify the RNA present in the sample. So it's involved different steps. First one is RNA extraction, which is the isolation of RNA from the cell or tissue. Second one is library preparation. In library preparation, we convert the RNA into complementary DNA and prepare it for sequencing. Third one is sequencing. Here we use different technologies such as NGS to read the cDNA and generate millions of short sequence. Then data analysis. In data analysis, we align the sequence to the reference genome to quantify the gene expression level and identify differentially expressed gene. Now, what is RNA-seq? RNA-seq basically provides us the comprehensive view of transcriptome and allow us for the detection of both known and novel transcript, alternative splicing event, and non-coding RNA. RNA-seq is widely used in cancer research, personalized medicine, and development biology. Now, in this project, we hypothesize that machine learning can efficiently differentiate and classify normal and tumor sample based on transcriptomic profile. For this purpose, first we will install all the required package and then we will load the libraries. So to download the data, and we will use the TCGA portal. So to download TCGA data with TCGA BioLink, we need to follow three main steps. First one is we have to query the TCGA database through R with the function GDC query. Then this will allow us to investigate the data available at the TCGA database. Then we need to download the TCGA data in our laptop or in our Google Colab. So in order to download the data, we need to install all these libraries. First we have to install all these packages. Once the installation is done, then we have to load all the required libraries. Everything is available in my GitHub profile. I will send you the link of my Google Colab notebook, which is in the GitHub. So you can use the same notebook to run transcriptomic analysis on your dataset. After installing all the libraries, here you have to load all the packages. So it will take some time. That's why I am not running this now. So here then you have to check all the available project available in TCGA. Here you can see there is a lot of projects available in TCGA. After this, here actually I am interested in colon adenocarcinoma dataset. So you can select any dataset in which you are interested. So when you will, if you want to get detail of all on the data that are deposit for TCGA adenocarcinoma, you can use this code. So here CODA represent the colon adenocarcinoma. You have to change the name of the cancer. So then here I am using this code to query all the transcriptomic profiling data from COAD project. This will give me the detail of all the transcriptomic data. Now here I can visualize the results that are present in TCGA. You can see all the results present in code TCGA. Then here I am using this code to query all the RNA-seq data from the colon adenocarcinoma project. So here you can visualize your result in more readable way. Now I want to see that to see the information of the sample present in this data. The information is present at a column sample type. So here you can use this code to see different sample type present in the cancer data set. So now I will convert the sample type to the factor and summarize using the summary table. I use this code to see this, and here you can see that there are 4 different sample type present in our colon adenocarcinoma data set, which is primary tumor, recurrent tumor, solid tissue, normal sample, and metastatics. Now here, as you can see that the metastatics and recurrent tumor are very less number. We have to remove this and we have to use the data set of only primary tumor and normal. So there are 41 control, solid tissue normal, and 481 are the cancer sample. We have to ignore the small classes of the recurrent solid tumor and metastatics. Therefore, we redo the query to check the sample type again. So I did here and I checked again. Here I use the same code to visualize the result. And now here you can see that I have primary tumor 481 and solid tissue normal 41. Now we prepared the download data. Now we have to download the data. So here we will upload the actual RNA-seq data into R. Both the clinical and expression data will be present in the object. So I run this code, and here you can see all the data present in the our colon cancer adenocarcinoma dataset. Now I will subset the data because we have huge data set, and if we use all the data set, then machine learning will take a lot of time. If you don't have any powerful machinery, then you can subset the data into small sample. Here I subset the primary and solid tumor into small data set, and then I run the code. Here you can see I get the small subset of the data. After this, you can run this to see all the detail of the data set. So here I run, you can see how many dead patients are there, non-reported cases, primary tumor, male, female, you can see everything. Now here we can check the dimension at the head of the subset RNA-seq by using this code. Then we have to save the file so that we can use our file to run the whole pipeline. So I save my data in code2data.rds. Now to perform the overall survival analysis, we will run this code on our code2data. Here you can see the survival plot. Now to generate the tumor stage specific survival curve, you have to run this code. This will show you different stages of the tumor: stage 1, stage 2A, 3A, 3B, stage 4. Then this code is for the survival analysis. Here you can see the survival analysis with different stages. Now RNA-seq analysis. Here we created a pipeline function for running a lemma package. So here in this you can use the code2 dataset and run the lemma package. You can also decide which one of the value of your conditional variable is going to be your reference group. So here I run the lemma package, I filter the data, normalize the data, and then fit the model, and then this will show you the top genes. So this function basically return a list with 3 different objects: a complex object which is from the running womb, and then actually this will basically show you the 50 differentially expressed gene which is sorted by the p-value. So here I will run the differential express analysis, which will compare the primary tumor sample and solid tumor sample. So you can run this code to see this. So here now we save the file as we just uploaded. We just uploaded the file This file we can use later as well. Now here we perform the principal component analysis to see the distribution and discrimination of the data. Here you just have to use this code and to perform the principal component analysis. Okay, then here we plotted different PCA based on the different features. For example, gender, primary diagnosis, pirate treatment, ethnicity, pepper tumor state. So these are the PCAs. Here in the PCAs, you can see in the PCS plot the data are not mixed, and moreover you can see that the sample type, the data are labeled and not mixed. So we can use the sample type for further analyzing the cancer data set. And here we will use supervised machine learning model for classification purpose. Using machine learning for the classification purpose, first we have to pre-process our data and divide the data into two group, training and testing data set. Okay, then here retain only a small subset of gene. Then we convert the definition into factor by using this code. You just have to use same code but only use the different cancer type. Then transpose and make it into a matrix, then convert the definition into factor. Then we will split the data into two different data sets. First one is training set and second one is test data set. We perform all this to pre-process the data. Here we are converting the data into training and testing data set. Make sure that 75% of our data will be our training data set and remaining 25% will be our testing data set. So we divide the data set into training and testing. Then data proportion table. This will show you the percentage of your training and testing. Here you can see that the 75% is our training and 25% is our testing data set. So here you will see all the available model that we can use machine learning model then we can use for our data set. So here I am using the ElasticNet model, which is a generalized linear model that combines the best of the two models, that is lasso and regression. So first we will train the model on the training dataset using the cross validation. Then we will evaluate our model using test set. Then this will show the performance of our model. Then we will see the statistics. Here you can see the sensitivity is 1, specificity is 1, precision is 1, which means that our model is giving us 100% accurate result. Now we will identify the differentially expressed gene from this dataset. You can run this code to see the differentially expressed gene. These are the ensemble IDs of differentially expressed gene. Here we found 139 relevant gene. Now we will compare the gene from the lemma and ElasticNet package to see how many genes are similar. So we run this code and here you can see 33 genes were similar in both the lemma and the ElasticNet. Now we can perform the enrichment analysis, and here you can see here you can run this code to see about these genes, to see the more information of these genes. Then here you can perform again the differentially express analysis. We use DE. Here we are using differentially express and perform differentially express analysis, and we are using differentially expressed gene from the ElasticNet and compare the top hit gene from the lemma. So here this heat map. In the heat map, the blue color show the top gene from lemma, and the top bar represent the primary tumor sample was represented in red color, and normal sample was represented in the black color. Here you can see. So then we will perform the hierarchical clustering by using hclust function, and then we will divide the tree into two groups. Upregulated, we will find the upregulated gene in the solid tumor sample and upregulated in the primary. So I run this code. Here you can see the cluster. We divided into two clusters. Then here you can see the upregulated genes, and then here to get the, then here if you want to get the common gene name of the first row, you can use this. Then here we created a variable, Boolean variable, that has that will show has true for dead patient and false for. Then here this code you will use to perform the survival analysis, and finally this code we are using to find the distribution of the differentially expressed gene. This box plot basically shows you the distribution of the differentially expressed gene, and here the blue color is for the solid tumor tissue and red color is for the primary tissue. So here if you want to find the upregulated gene in normal tumor, this code basically show you the upregulated genes in the normal tumor group, and this will show you the upregulated gene in the primary tumor group. And here finally we perform the survival analysis. So this is the overall result of the survival analysis. And then here we divide the patient into two groups, upregulated and downregulated. If the patient expression is greater or equal to the median, we put it among the top regulated, otherwise among downregulated. So here we check the expression of the gene and also it influences on prognosis. So here we took all the expression value in the disease sample and then took the median after them. So here the patient with the expression greater than median, we add these in the upregulated, whereas other in the downregulated. So this is the p-value of all the genes that are upregulated and downregulated. Here I uploaded the necessary libraries to plot the survival analysis. This plot basically show the overall survival analysis. This pink color basically show the downregulated gene, and this blue color basically show the upregulated gene. So this survival plot basically represents the difference in the survival probability between the upregulated and downregulated gene, and this plot basically include the p-value confidence interval along with the risk table for more context. So this is all about how we can perform the RNA-seq analysis in Google Colab. If you have any question, please leave your question on the comments below, and thank you for watching this video. In the next video, I will explain how we can use other machine learning model like a random forest sport vector machine for the classification of the RNA-seq data, or for the classification of the tumor sample by using these models. Thank you very much for watching this video.